Thank you for joining us again for a bite out of science. This evening or afternoon, we'll be taking a bite out of cancer. And so thank you for joining us again for a wonderful webinar this afternoon. So we'll start off with a little bit about the Center for Excellence in Education. So we were founded in 1983 by Joanne DeGennaro and Admiral Rickover, the father of the nuclear navy, and civilian uses of nuclear power, with a mission to nurture high school and university scholars to STEMs of excellence and leadership in STEM. So we currently have 4 programs that work under these wonderful initiatives, and these programs are for teachers and students. So we have our Research Science Institute, which is RSI, USA Biolympiad, which is USABO, Teacher Enrichment Program, which you are participating in tonight. And our newest program is STEM Lyceums, that focuses on helping high school students to gain more information in careers of STEM. So with our Research Science Institute, this is for it's a highly competitive 6-week summer science and engineering program that is open to rising high school seniors, and juniors are eligible to apply. That application will actually be opening within the next month. So please be on the lookout for that application opening so you can inform your students to please apply. And it is great for them to know that RSI is a cost-free summer science and engineering program that combines on-campus coursework in scientific theory with off-campus work in science and technology research. Our USABO will also be opening soon for applications, and this is a premier biology competition for high school students in the United States. With being that, it provides the motivation, curricular resources, and skills training to take them beyond their classroom experience to a level of international competitiveness. And since CEE began administering the USABO in 2003, every member of the USA Bio-Olympiad team has medaled at the International Bio-Olympiad. Our Teacher Enrichment Program, which you so wonderfully are participating in tonight, provides opportunities to middle and high school teachers to connect with leading experts from industry and academia, explore cutting-edge research and development, and make meaningful professional links with direct benefits for you and your students. And as I stated earlier, our STEM Lyceum is our newest initiative here at CEE, and it's a virtual club for high school students to engage in discussions and exploration of STEM concepts and career pathways. So this afternoon, we have a wonderful speaker. Dr. Sharnise Mitchell is a research program manager in chemical biology and therapeutics at St. Jude Children's Research Hospital. She received her doctorate in philosophy and biochemistry and molecular biology from The Ohio State University. And with over 15 years of research experience, she is going to give us a wealth of knowledge tonight of how she got to her position and how you can help your students to do the same. And she's also encouraging to ask questions throughout her presentation. So please, let's make this more of a dialogue tonight than just a Q&A at the end. So now I will turn it over to Dr. Mitchell.
My name is Sharnise Mitchell, and I am currently a research program manager here at St. Jude Children's Research Hospital in the Department of Chemical Biology and Therapeutics. I'd like to start with an agenda, so I'll give you a little bit of idea of what we're going to talk about today. The first is going to be about me, my education, some of the fellowships that I participated in, and how that kind of catapulted my career and where I am currently at now at St. Jude, and some of the things that we work on St. Jude and what my day-to-day looks like. I began studies at Tennessee State University in Nashville, Tennessee, where I earned a Bachelor's of Science degree in chemistry with a minor in biochemistry. During my matriculation at Tennessee State University, I was one of the students fortunate enough to participate in a government-funded program called the MARC program, which is Minority Access to Research Careers. And I truly credit this with all of my success moving forward, because it gave me an opportunity to explore an area that I knew nothing about prior to going to college. And honestly, I had no idea what I was going to major in when I got there. But through summer internships, I spent a wealth of my time really learning about different topics and subjects in science. And it really allowed me to hone in on what it was that I was most interested in and what kind of tickled my fancy, what made me excited. The structure of the program was that you spent time as a summer intern at 2 universities over the summer. However, during the academic school year, we were researchers in a laboratory on campus. So my 2 summer internships, the first was at Purdue University, and the second was at Penn State University in State College. I learned so much from those experiences, and probably the number one thing that I learned was that I wanted to continue doing it, that I really loved research, and it was something that I was passionate about. Another huge benefit from being in the MARC program was that I was a published author before I'd even graduated college. So the paper here where I was second author in environmental research was actually work that I had done as a sophomore and junior while I was in school. And before I had even made it to graduate school, I had already published a first author paper. And in some programs, that is actually the requirement for PhD graduation. And so having access to those scientific programs and those laboratories and the experiences that I got, I was making really great strides towards my career. Moving on to graduate school, I interviewed with a lot of different graduate schools, and ultimately, I chose The Ohio State University for a number of reasons, but primarily because I felt like it was kind of like the scientist's wonderland in the sense that there were so many different options and opportunities. I actually went into an interdisciplinary biochemistry program, which allowed me to rotate in a lot of different subject matters. It allowed me to learn about a lot of different laboratories and some of the research topics that they were working on. And because I was so eager to kind of begin my work, I actually began my summer rotations before the first year of graduate school. And this was truly an advantage that I didn't really know about. I kind of wanted to get away from home. And so I was like, well, can I start early? And they were gracious enough to allow me to do so. But it kind of gave me a leg up on all of my classmates coming that first year because whereas I was learning a new environment, living in a new city, all of these things that I would have to learn kind of like at the beginning of the school year while now starting graduate level classes, I was able to tackle some of those things before the school year had started. And that was through doing some of my summer rotations before the first day of school. While I was there as a second year, I applied to an NIH Health Service. It was the Ruth Kirstein Health Service Award, which was a predoctoral fellowship that actually supported my work through the entire time that I was in graduate school. And some of the fruits of my labor were 2 publications, one in nucleic acid research, as you can see on the right, and the other in ACS publications. And so I really learned a lot from graduate school. My studies were purely biochemistry based, where I was looking at in bacterial. So let me back up and explain the lab that I ended up joining. I ended up joining a lab. So I had classical training in chemistry, but I learned that I was really interested in biochemistry, and thus I went into a biochemistry program. But as I was in that program, I rotated through a number of different labs and ended up really liking a lab that was genetics based. I ended up training under a geneticist where we studied the genetics of bacteria. And one of the phenomenons in bacteria, and this was some time ago, but it was I worked with the person who discovered what's called riboswitches, and she basically identified these genetic regulatory mechanisms that sense the environmental cues and actually modulates gene expression based on what is going on in the environment of the bacterial cell. The riboswitch that I was focused on was the L box, and it stands for lysine. So what it would do was the cell would make RNA, and the RNA would form into a secondary or tertiary structure that could then bind the lysine molecules. And so my studies were really trying to understand what in the RNA actually recognized the very different little pieces of a single lysine amino acid. And that was basically what the nucleic acids research paper published, was analysis of lysine recognition and specificity in Bacillus subtilis L-box fiber switch. This work also was translated over into some other systems, and one of those was mechanisms of resistance and how understanding that information could also target other processes in the cell, such as translation. Although it was the transcriptional regulation that was occurring in the L box, it could also impact translation, and that is what the subject of the other publication in ACS Publications is about. So I want to start by moving on. So after I graduated grad school, well, actually prior to graduation, we had lots of recruiters coming around, and I had a pretty good idea that I wanted to go ahead and continue a postdoc, but it was an opportunity for me to kind of pivot and change gears. And so my host organism and the organism that I had been working in was bacteria all of this time during graduate school, that I thought it might be a good idea to kind of jump organisms, jump hosts, and make my way over to humans. And so I'm going to share with you in this next slide a video that I watched prior to coming here to St. Jude. And I really would like for you guys to take a look at it and use some of the information and how it made you feel. I'll let you watch it first and then we can talk about it later, but hopefully you can use some of the ideas and the tools around it to inspire your students to go for what they're passionate about, because that's what I got after watching this video.
You're one of the rare few, the ones who have found that reason, whose calling has set them on a path to spark something greater than themselves. You've dedicated your life to reaching that breakthrough, that moment your calling becomes someone else's chance at life. At St. Jude, we know what it takes to answer a calling. It takes a team dedicated to advancing cures and saving children on a global scale, pairing the brightest minds from around the world with the most advanced technology, and doing it all while sharing our findings freely and giving our researchers everything they need. When it comes to pediatric disease, we know there's no time to waste waiting for funding or resources. If something is needed, we don't wait for someone else to create it. We create it ourselves. From installing the world's most powerful superconducting NMR spectrometer to building and maintaining libraries with over 1 million registered compounds and fragments, St. Jude is home to countless on-campus, one-of-a-kind innovations, all dedicated to streamlining the research process. Our staff has access to everything they need to reach our shared goals. We've heard our calling. And when you're ready to answer yours, we'll be here asking, what do you need?
Dr. Mitchell.
Yes.
We do have one question,
Okay.
kind of where we are right now.
Sure.
What encouraged you to make that pivot from bacteria to humans? Before you saw this video and the recruiters, what made you want to make that pivot?
So part of graduate school, you sit in a lot of seminars and you learn a lot about the different systems and mechanisms and organisms. And although bacteria and what they do and even how they affect humans is truly important, I really felt that I wanted a greater impact. I wanted really to see the fruits of my labor, or for my work to go towards something much larger than me. And although bacteria are definitely an interesting organism and there's lots of work to be done within them, I really wanted my work to apply to the human cause. And so that was why I made the switch. Any other questions? Thank you for asking.
We do have a few. I don't know. Do you want to continue to go?
No, it's perfectly fine.
Okay. There was one of, how did you discover the MARC program, and was that a program you applied to prior to entering college?
Yes. So I actually discovered the MARC program through my roommate in college, who was a psychology major. And she had learned of it through maybe some mutual friends of hers. I had not known of it before then, but it was a program that was also interdisciplinary in that they accepted students with the different majors. So there were some of us with chemistry background, there were some of us with biology background, there were some of us with psychology backgrounds. And I had no idea about it prior to actually going to college. I did participate in other summer internship programs. There was a program at the University of Tennessee, Memphis that I participated in as like maybe a sophomore in high school.
Mm-hmm.
And it was called SSEP. It was such a long time ago. I can't remember what they stand for, but it was kind of a summer science enrichment program, SSEP. Okay, that's what it was, summer science enrichment program. And so I knew those things existed. I just didn't know where to find them. And having the exposure kind of early on gave me the idea that, okay, there are these things out here. And one thing that I guess I did not mention was that for every program that I was in, one of the major incentives was we got stipends for everything that we did. And so part of my motivation was as a high school student, I wanted to buy the clothes that I wanted to buy. You know, I wanted to have my own finances and my own money. And I kind of used that thinking to push me all the way through graduate school because I was always in a program that paid a stipend and helped support my way of life or living ultimately. So I never, I don't feel that I struggled as I could have having been in graduate school because normal graduate school stipends aren't the best, but being having a fellowship and understanding that for your kind of educational prowess, you can also support yourself was something that I learned early on, and that is why I did it.
And we have one more for the moment, and it said, how did you become comfortable in your scientific writing?
A lot of trial and error. Track changes. I can't always say that I'm even comfortable now, but I'm comfortable with the process and understanding that your first draft is not always the final, and not even your 10th draft is the final, but constantly reconsidering what you're saying and trying to misinterpret what you're saying so that you can be very concise and very specific about the work that you're conveying to the world, because ultimately people from all different backgrounds and all different subject matters will read your work. And so I'm really interrogating what you're doing and what you're writing is part of that process. And so being comfortable with that, I think, is helps support the work that I do even now, even today.
Awesome. Well, that's all we have for right now.
Wonderful. So I showed that video just because I was moved when I saw the video as a graduate student. And I knew that, again, my calling was bigger than the genetics of bacteria. And so that is how I ended up at St. Okay. That is how I ended up at St. Jude Children's Research Hospital. This is a view of our campus. I know you see it all the time on the television, but it's really near and dear to me because it's something that I see every single day. And I feel fortunate to be in a position to do so. This is kind of a sneak peek of the inside. This is kind of the merging of two buildings. One building, the building on the right, will house some of our patients, and the building on the left is actually a research center. And what's really unique about St. Jude is that we have both clinicians and scientists working together for the cause of childhood catastrophic diseases. I'd like to point out to you that the picture is from the bottom, but those buildings are usually 8 or 9 stories high. And what's really cool about what they've done here is that all of those little, this mural or this image that you see in all of its beautiful color is actually individual sequins that were placed on the walls. I do not recall how many are there, but it is in the millions. And on a bright and sunny day with the wind blowing, it is such a beautiful display of just color and artistry. And it's just one of the things that it is unique to St. Jude, but this happens all over our campus, and we really love to be the first to do the things that other people aren't doing. This is the building that I walk in every day, even today. To the left is the Danny Thomas Research Tower. And I don't know if you recall, but on that video, there was an aerial view of what looked like it was inside of a building. And what it actually was inside of the Danny Thomas Research Tower, which is if you're facing this, is to the left. And that building is where I started my postdoctoral fellowship, where I worked in the lab under a physician scientist who was studying acute megakaryoblast leukemia. I currently work now in the building to the right, and I'll discuss more about that later. So let's talk about my postdoctoral fellowship. It was in the Department of Oncology, and I want to kind of walk you guys through what a typical workday for a scientist looks like, at least for myself. So I usually have a plan. We always have a plan of what's next, what we're looking for, what the data is. So everything begins at the desk. We plan the day based on whatever our larger overarching umbrella plans are. For me, I choose to read over my protocols, especially to identify those reagents that need to thaw, that need to be at a certain temperature, that may need a little extra TLC before the experiment begins. I go to check to make sure that we have everything, because as you know, laboratories are, they're a team sport, so we're all in it together. We have to share things, and we definitely have to be prudent about our reagents that we're using, because after all, we are funded by donor dollars, and that is really important to us. We don't take it lightly. So I check my reagents, I prepare all of my safety items, so lab coat, goggles, and depending on where I am, or the year, masks, or if I'm working in the cell culture, we'll don a different type of PPE in order to keep not just us safe, but also to keep our experiments uncontaminated. And then I start my experiments, and those can be a variety of many different things. We typically, it was good to have had the experience in bacteria because a lot of the work translated as far as assays were concerned, but it was really the biochemistry background that really helped me to be able to troubleshoot a lot of different experiments and understand when something wasn't working based on knowing the chemistry of the assay. So not necessarily for my data or my results, but understanding the reactions that are occurring to yield those results, and knowing how to troubleshoot and modify them. Some of those things could be DNA extraction, cell culture, cell purification, protein purification, all of things that I learned having been in actual bacterial lab, because at one point in graduate school, we were actually purifying our own proteins for everything. And so that experience was really great because there are techniques and tips and tricks that transfer even when you're working with a different organism. And so I have some photos of what that looks like. And so this is what we call these mini preps. Basically, this is DNA that we are capturing from a particular cell type. That's actually not my hand. This is the stock image, but this is typically what the mini preps look like. And you can see at the bottom of the tube that there's a precipitate. And eventually the goal of this assay is to separate the DNA from all of the cellular debris, purify it in such a way that it can be run or analyzed. This is a larger example of that, and in some cases, this is it could be protein purification from those same types of cells. If you look into those 50 mil conicals, you'll see kind of like a little cylindrical device, and that's typically a filter. And that filter is used to capture protein or to capture DNA or whatever is being purified on a much larger scale that eventually is collected and put together for whatever the next ensuing assay is. This is just an example of one of my more recent publications from work. As I mentioned before, I work in a lab that studied acute megakaryoblast leukemia, and it affected young children, typically 2 years or and under. And the goal of this work was to identify or better understand the current standard of care and the chemotherapeutic treatments that were happening in the clinic, to identify ways that we can improve upon them or work to make them better. And so what you're looking at here are actual leukemia cells that were stained with antibodies that recognize two different proteins. And those two different proteins are lit up in two different colors. And so what we wanted to know was how those proteins change upon treatment with either bortezomib, which is a chemotherapeutic agent, or vorinostat. The real question is who's the biggest player, and the effects that we see, is it a result of bortezomib treatment, or is it a result of vorinostat treatment, and which of those combinations could be substituted to try and offer more effective treatment. And as you can see, the tubules, which is what you're looking at, are very striated, and you can see the spindles at the end of the cell. But once you add vorinostat, under any circumstances, whether it's vorinostat alone or bortezomib and vorinostat together, you can see that we've lost some of that spindle formation, and now we're looking at more aggregated protein instead of the uniquely formed protein as what we were seeing before. And that work was published in a Nature Communications paper that you can see above, discussing proteasome inhibition.
We do have one question in the chat.
Sure.
And it was saying, what did you learn in high school that benefited you along the way, from college to grad school, your doctoral program, and now within your current research at St. Jude?
Absolutely. There's one moment in high school that I always look back on because it impacts me still to this day. And I was in what was called a Medical Laboratory 2 course, and we had taken a quiz, and the quiz was it was not mathematics, but on different items in the laboratory, basically. And I was upset because I had gotten an answer correct, but it was marked wrong. And when I asked the teacher why was it that the answer was wrong, she told me it was because I did not have units behind my answer. And so I cannot, to this day, look at a number and not ask what the unit is associated with it if it's not already predetermined. And sometimes I write it completely unnecessary. But what it taught me was the value of details and how it is so important to be detail oriented in everything that you do, that it's not enough just to do it. It's more to do it well. And so that is kind of what my takeaway was as a high school student, was that everything that I do, I shouldn't just do it to get it done, do it for completion, but I should do it well. I hope that answers the question.
I think so. That was great.
So if we don't have any further questions, I want to switch gears and talk a little bit more about another pivot that I made. So the lab that I was working in, the leukemia lab, had kind of gotten big and had grown and was relocating. And the leukemia lab was relocating to Stanford University in California. And wonderful place. Sounds like a really, really great place. But I was tasked with a decision on whether I moved to Stanford University or stay at St. Jude. And to be honest, I'd come to St. Jude because of me wanting to impact the world, essentially, wanting to impact science and health and human disease. And so I felt like it was best for me to stay at St. Jude. And what I ended up doing was moving somewhat away from the lab and taking it was actually a project coordinator position at the time. This was during the pandemic, but it was a project coordinator position in the Department of Chemical Biology and Therapeutics. And I just want to give you a little bit of background on chemical biology and therapeutics. And our mission in our department is to discover new chemical probes that can be used to study disease biology related to catastrophic pediatric illnesses. So when you hear that mission, it sounds like the perfect marriage between my training as a scientist and my education as a chemist. And so I thought it was a really great opportunity for me to pull more of my skill set and effectively use it in my day-to-day work. I did not mention this before, but what I will say is, although these are beautiful stock images, I knew 70% of the people that were in the first video, and who you're looking at right now is Mariana. She works in our compound management department. I work very closely with her. And so as you look at these images, I will try and point out to you the people that I work with. Some of them sit right behind me or right next to me, just to reiterate that science is a team sport and not one person can do it all. So how do we strive to reach that mission? And what we do is we perform research that employs state-of-the-art techniques, including high-throughput, fragment, virtual, and high-content screening, structure-based design, synthetic natural products, and parallel chemistry and pharmacology. And I know that was a mouthful, but I will walk you through and give you an example of what that looks like for us. So how does a project manager or program manager at this point, how do I impact the now science that I've kind of stepped back from to do? And I think what we look at science and we look at experiments as isolated events, but in truth, they are really long dances, so to speak. And it's what we call the collaborative project process, because again, science is a team sport, and no one person can do it all. And so there are certain steps that we go through for our projects. And as a program manager or as a project manager, I'm involved in many more steps than what I would have been just as a scientist at the bench. And what's nice is that I'm able to use the education that I have to help the teams make informed decisions about the work that they are embarking upon. So typically a project starts with the design. The collaborator meets with project managers and assay development scientists, and this is really a feasibility check. Is this reasonable? Is it something we can do? Can we afford to do it? Will it yield the results that we're expecting? And sometimes those discussions can go many different ways, but typically it kind of lays down the framework for what a scientific project looks like for us at St. Jude. And then we go, and our second step is resource allocation. We look and we say, okay, this project is a go, it's approved. Now let's figure out what kind of resources we need, what the budget looks like, what are our FTEs, who's going to be employed, who's going to actually be the hands on this project. Those things are determined, and the project manager is involved in that case too, because especially when we're considering interdisciplinary teams where we may have a chemist and a biologist and a structural biologist, and project managers, as well as many other people that work on it. We have computational chemists or computational scientists that will work on the same project. The project manager's role is to have the ability to, in some instances, liaise between those teams, but as well as translate what the work looks like for them to make it palatable for everyone involved. If we are screening a library, which when you think of a library, we're talking about a chemical library. So for as many books as there are in a chemical library, there are just as many chemicals, if not more, here at St. Jude. And I'll talk to you guys about that here. But there's a single team that manages that, and that's called compound management. We select the libraries. We talk about what the investigators want to do, and those libraries are selected and formatted or prepped for the assay. The scientists go and they optimize the assay, they optimize the performance, and they establish automation compatible protocols. So protocols that will allow them to do high throughput screening. And then there's the high throughput screening. And what we mean by that is instead of a single assay where you saw a picture of one person manually isolating DNA from, let's say, 15, 16, 17 different samples, we're talking about robotics that will do hundreds of thousands in a single run if necessary. And that's the image that you see to the left. Here, there's a plate, and this is an arm, and this is a part of a really large instrument. And this arm is either rotating this plate or moving this plate from left to right as a part of the assay that it is running. Once the screening has occurred and the robots have done their work, there are secondary assays that are performed to validate the leads from those screens. And then the project managers come in here because if there's chemistry to support desired, we go and we communicate with those chemists and say, hey, this is what's happening in this project. We may need your help on optimization, or tell us what your thoughts are on what that looks like. And that is kind of how the collaborative screening process goes here at CBT. Also, there is the project manager's favorite part, which is the project conclusion, because the whole point is to get the job done. And that's where lead compounds are transferred to the collaborating lab for follow-up in model systems. And I feel like I just walked through this a little bit, but the typical workday for a project manager typically starts the week before. So we're always planning for next week, sometimes next month, sometimes next year, sometimes the next 5 years. But on a typical day, I've already reviewed the week that I'm in, and I'm reviewing the next week. I'm setting meeting agendas. I'm preparing schedules for the day. I'm writing down those investigators or scientists that I need to talk to. I'm communicating with those project teams to let them know where we are on things or what needs to happen. I'm planning reagent transfers. So sometimes everything doesn't stay within St. Jude. We're very collaborative, even with other institutions. So we send things out. Things come to us, all for the purpose of research. And then I spend a lot of time scheduling communication meetings. And this is very important because this is really the meeting where the scientists update on the work that they're doing, the progress that they made, the data that they've acquired. And this is really what keeps the projects moving forward. I monitor experiment progress. I help aggregate experimental results. I update communications pages that are more static, meaning that people just come and visit them instead of us pushing communications out to them. And I overall ensure that our scientists have the tools required for their experiments. So I talked to you guys about compound management and them being part of one of those spokes in the process. And we have a center called the Brooks Life Sciences Sample Store. We call it the BLISS. We have a habit with our robots of naming them. One of the really cool things about our department, because we have so much automation, is we have a naming contest for our patients. And our patients actually get an opportunity to name the instruments that are so important in the work that we're doing, and ultimately the work that potentially could directly impact them. So BLISS, better known as Jarvis, can store between 70,000 to 1 million compounds. And what you're looking at is the corridor within that sample store. And so on the left and right, these little blue items are plates, and those plates can have 384 wells in them, sometimes 1,536 wells in them, but they're stored up and down this wall. And there's a robot here who is in motion right now, so you can't really see him as well. I think there was also an image on the initial video that I showed, but the robot moves back and forth through this corridor and pulls the plates for us when we're requesting compounds. Currently, St. Jude houses about 750,000 compounds that are freely available to our investigators to use for their scientific research. This is another image of our sample storage module without Jarvis moving back and forth. And you can see that there's a different type of plate here on the right, but that's what those little white specks are. And the barcoding is what's used to identify what's in the plate and where it is. And as you can see on the left, this is what we call our transfer. The instrument will come down and there is a plate here, and that plate will line up with this here and transfer those compounds out for our compound management team to then take and reformat for our investigators. Here is another image of Mariana, which we've seen before, but this is also Dwayne on the left, and he is a part of our high throughput biosciences team. He sits right here. And unfortunately, I kicked him out of the office for today's presentation, but he is very instrumental. And this is a much better view of the two arms that are used to pick up the plate. And this is the robotic arm that will then rotate 360 degrees to go left and right. I'll show you in a little bit kind of an overall view of that to give you some perspective on what this looks like. But the key to this slide is collaboration. Compound management needs high throughput biosciences for their requests. They are the ones who are using these compounds, and high throughput biosciences needs compound management because they need the compounds to do the studies and to do their screening projects. This is Widget. This is another instrument. You can see here that there is a Staubli arm that rotates. It rotates in a lot of different angles, but it basically effectively picks up plates from this tray carrier here, and we'll put it in either this plate sealer. There's an incubator to the right here, or a plate reader. And this is just an example of one of the programs. I believe these are undergraduate students that were coming and visiting, and Dwayne was giving them a talk about Widget and what Widget does. And Widget was also named by a St. Jude patient. And lastly, I guess I have another image of this is a 1536 plate where you get an opportunity to kind of see down into the wells. They're very small, but you can imagine that hundreds of these plates can offer really, really rich data for screening projects if the assays are compatible and correct. And so I have another video, and this video is the aerial view of the instrumentation that you've seen before. So to give you some perspective, that robotic arm that I was speaking about is right here. There are plate carriers here, and there's lots of other instrumentation, but this is the track that the robotic arm will move plates from instruments on this side, move them over to run tracks onto this side into either the AmbiStore, which is what this is called. This is the Prime, which is our newest robot in the High Throughput Bioscientist Center, or any of this echo machinery that is on the right. And I guess I have to change my pointer. Oh, where is it? Okay, let's see. It won't allow me to.
I think if you do the visible, yeah.
Got it. Okay, hopefully you guys get the idea. It's just the same loop running over and over again. This was a lapse of over a single work day in all of the work that they were doing in the High Throughput Biosciences Center.
Dr. Mitchell.
Yes.
How much do you use AI in your research labs?
So we do use AI, and before AI was really popular, it for us was called machine learning. And so basically what we did was, it's also being used in scientific research as well, where it's used significantly in some cases to predict the feasibility of experiments, to share ideas about potential targets or potential protein structures, especially for those things that are just unattainable or unavailable to us. It's definitely being used. And I just imagine that it's going to explode now, especially with the popularity and the adoption of it kind of by the world. So we do use it a good amount, but there's still nothing like, although the robotics and machine learning and AI instrumentation is important, there's still nothing like the human brain.
Always.
Excellent question. Okay, here we go. Did I do? Okay. So this is an image of the wall right outside of that High Throughput Biosciences Center. And I want to focus on one picture, and that's the picture in the center. And this is of a St. Jude patient named Roman. And Roman was the name contest winner for a robot called Tobar. And if you think about it, It's actually robot spelled backwards. And so the other images are images of St. Jude patients. We had Clifford the big red dog. We have lots of different names for our instrumentation, but it just highlights in how the patients are the center at everything that we do. So I want to give one example of let me look at the time. Okay, I'll move much faster. Sorry. This is an example of a screening project called the Cisplatin Protection Project. And basically, cisplatin is a chemotherapy drug that's used in a variety of cancers. And basically, it attacks the tumors specifically and is effective to treat a number of different cancers. However, cisplatin also impacts hearing, and it was identified that a loss of hearing was an off-target effect of cisplatin. And so the goal of the screening project was to make an effort to identify another drug that would protect the cells in the ear that was causing this hearing loss impacted by cisplatin. So the primary screening mode was cisplatin with isolated inner ear cells and a number of different compounds. They're screening for this compound and will select for survival of the inner ear cells. There was a secondary screening module where instead of using cisplatin protection, we were just trying to identify compounds that could be selected for survival of the inner ear cells. And this is an example, after a long series of experiments, a project for which I was not actually on, but this is an example of the data that was from that screen. And basically the enzyme that they were measuring was called caspase, and they were looking at caspase activity. So when the activity was low, it was a direct correlation to lower cell death. Every dot is a color-coded well, and you can see that the gray are sample compounds, the green are media only, the red are cisplatin only. And the compounds of interest are those that produced the lowest amount of caspase activity. Those hits were carried on for further testing. And you can see here that in comparison, you can show survival of the cells in 2 with dorafinib, which was one of the lead compounds that was identified. And so lastly, I want to finish with our motivation. And I know I had 3 videos, but this is the last one. And hopefully it really explains to you or helps you guys understand why we do what we do.
My name is Chloe. This is my brother Roman. He's a patient at St. Jude. My name is Roman, and I want to be an inventor.
If you were an inventor, what would you make?
A robot, new medicines for St. Jude, flying cars, a new kind of train, and a new chemo machine. I'm making a new robot. There's some energy source. This medicine is going into this tube so it can be transported to St. Jude, and I'm holding a chemical I just created.
Why do you want to be an inventor?
Just to make the world a better place.
And with that,
My name is
I'm sorry, I do this every time.
This is my brother Roman.
And with that, I'll take any questions, but my last take-home message is that you can do it too. You don't have to be at St. Jude to inspire children to do research. These are just some examples or websites that may help, but I'm here to answer any additional questions that you guys may have.
Okay, we do just have a few. One comment was saying they wanted to thank you for sharing the collaborative project process because it gave them an idea on how to create a new lab in their classroom.
Wonderful.
So they said they wanted to use that to kind of do a new way of teaching the scientific method. So
That's
that
awesome.
was a great way. And another question was, does St. Jude have any opportunities for teachers or high school students that you know of?
I do believe that there are opportunities for high school students. It would be best to reach out to our cancer education and outreach program because they will have the details and the step-by-step information on what needs to happen. Anika would be your best contact for that.
Okay. And so if anyone would like that, I can share that information for you for outreach. And our very last question was they were interested in what is the process of protein purification?
It depends on the protein. So I can give you the example from when I was in graduate school. We were purifying the RNA polymerase, which is and it was actually RNA polymerase holoenzyme, which in bacteria is only a few proteins as compared to the number of different proteins that's required as you start to get into other higher level organisms. So the number of proteins will increase as the complexity of the organism increase. But basically, RNA polymerase is responsible for producing RNA from the DNA that is in your cells. And as you know, RNA is used to translate proteins, and proteins are basically what they call the workhorse of the cell. They're responsible for a lot of the action and activity that happens in the cell. But RNA polymerase has to be functional and work to make RNA. I mentioned that I was studying riboswitches, which are secondary or tertiary three-dimensional RNA structures. And in order for us to make those structures in a tube or in a cell, we had to have a pure system. We altered a different bacterial species to harbor what we call a His tag. And what the tag does is it has a really high affinity effector or high affinity binder that will allow for us to use that binder on a column. And what we will do is we will grow up tons and tons and vats of this cell with this modified RNA polymerase holoenzyme. We will then lyse the cells to basically break them open and then pour that lysate over the column. And what will happen is the His-tagged protein will be retained on the column and everything else will be washed through. This is a process that has to happen. Typically, protein purification has to happen at 3 and 4 degrees because you can imagine that proteins are not stable always at room temperature. Not all of them. There are some.
Mm-hmm.
But you wash them over that column, you purify that protein, and then you have to identify where your active fraction is. And so then there's a fractionation process that occurs. You pull your active fraction, you pull them together, and now you have a functional RNA polymerase that, if given the right substrates, will make RNA in a tube.
Well, thank you so much for your wonderful presentation, all
thank you.
information. And I want to thank all of our wonderful teachers for joining on. As I said earlier, if you have any questions about any research or outreach from St. Jude, please contact me and I can share that information with you. But we want to thank you all for joining us again, and thank you to our sponsors, and we hope to see you on our next webinars.